for joining us. We are delighted to have a fantastic guest today. My name is Christina Iptoma, and I am mom to OsteoAngel Dylan and Director of Scientific Programs at MIB Agents. And today on OsteoBites, we are talking with Dr. Janet Yoon about a Y-mAbs Therapeutics first-in-human phase 1 trial for patients with recurrent or refractory metastatic solid tumors known to express GD2. Thanks so much, Dr. Yoon, for joining us on OsteoBites today. We're thrilled to have you. And thank you to our panelists, Vicki and Walker, for joining us today. Vicki is our Junior Advisory Board President, and Walker is our Vice President, and they're both osteo warriors. And a little bit more about our guest today. Dr. Janet Yoon is a director of the Pediatric Musculoskeletal Tumor Program at City of Hope. She is actively involved in clinical trials as the site principal investigator of the Children's Oncology Group and as a co-leader of the Pediatric Disease Team. And prior to joining City of Hope in August 2022, she was the director of the Solid Tumor Program at Rady Children's Hospital in San Diego. And today she's going to provide an overview of a phase 1 trial with GD2-SADA 177Lu-DOTA drug complex in patients with recurrent or refractory metastatic solid tumors known to express GD2. There are lots of letters and numbers in there, so I'm sure Dr. Yoon is going to break that all down for us, all that alphabet soup. Welcome, Dr. Yoon, and welcome everyone joining us today. Please feel free to add any questions you have for Dr. Yoon to the Q&A as we go. And before we get started, just a couple announcements and reminders, because we can't stop talking about Factor, which is our annual conference, June 22nd to 24th in Atlanta. The agenda is up on our website, so you can see our amazing speakers and panelists. We have 50 speakers this year, which is amazing. And Dr. Yoon is actually going to be there too. So if you're interested in the study and want to learn more about it, come to Atlanta and you can talk to her in person. We will have 6 scientific panels covering biomarkers for risk stratification, preclinical models, comparative oncology, local control, immunotherapies, and molecularly targeted therapies. And we're going to have 3 interactive smaller breakout discussion groups this year. One is going to be focused on comparative oncology and moving from canine trials to pediatric trials. A second discussion group is going to be on leveraging computational biology to accelerate osteosarcoma discovery. And then our third discussion group is going to be about turning pain into power, patients with osteosarcoma who enable research, and how we can best engage the patient community to power research with their data and tissue samples. Again, Dr. Yoon's going to be there. You're going to learn more about the study today. You can ask more questions there. And if that isn't a big enough draw, there will also be yoga with bunny rabbits and sound healing for those families who aren't attending the scientific sessions. So there truly is something for everyone. One other plug, our May issue of our monthly newsletter, Connected Issue, just went out yesterday. And if you aren't already receiving it, just go to our website and click the join button, and then you'll get it delivered in your inbox every month. And you can access past issues on our website in the About Us section. All right. How about Vicki? I'll hand it over to you to introduce yourself.
Yeah. Hi, everyone. My name is Vicki Hoy, and I'm the president of the Junior Advisory Board, as Christina said, and an osteo warrior. I'm coming up on a 1-year NED in 2 weeks, which I'm very excited about. But handing it off to you, Walker.
As Christina said, I'm the Junior Advisory Board Vice President, and then I am about 4 years removed from treatment, so that's pretty exciting. And then Dr. Yoon, you can take it from there.
Thank you very much. I'm very happy to be here, and I'm quite honored to be speaking today. I will be presenting the Y-mAbs Therapeutics Trial 1001. It's investigating GD2-SADA complex coupled with 177Lu-DOTA in patients with recurrent or refractory metastatic solid tumors known to express GD2, which include small cell lung cancer, sarcoma, and malignant melanoma. This is an investigational product, and the efficacy and safety have not been established by any health authorities, so this is not approved therapy. So by way of introduction of osteosarcoma, which I don't need to introduce to this group, it is the most common type of cancer arising in bones, encompassing 2/3 of all bone cancer cases. It's the third most common childhood malignancy, mostly affecting adolescents. There's been no significant changes to neoadjuvant treatment approach over the past 3 decades. The 5-year survival for those with non-metastatic disease is about 65 to 70%, and the 5-year survival for patients with metastatic disease is about 20 to 30%. There is a definite need for new therapies to address patients with metastatic, recurrent, or refractory disease. So the GD2 antigen, which is the target in this trial, it's a ganglioside, which is a carbohydrate containing sphingolipid that is widely expressed in normal tissues. It's involved in cell recognition and membrane-bound signaling protein regulation. The ganglioside, GD2, has limited expression in normal tissues, but it is expressed on CNS cells, peripheral nerves, skin melanocytes, and mesenchymal stromal cells. It's been shown to be highly expressed in cancers of neuroectodermal origin, which includes melanomas, Ewing sarcomas, osteosarcomas, and soft tissue sarcomas. GD2 is a valid target in oncology through the work of our neuroblastoma colleagues. FDA has approved 2 monoclonal antibodies directed towards GD2, dinutuximab and naxitamab, for the treatment of neuroblastoma. Anti-GD2 therapy has been investigated in osteosarcoma. GD2 is known to be highly expressed in osteosarcoma, being present in over 70% of osteosarcoma tumor specimens. Roth et al. In Cancer 2014 demonstrated that relapsed osteosarcoma specimens had higher GD2 expression compared to samples at initial biopsy and at time of definitive surgery. Anti-GD2 therapy induces antibody-dependent cellular cytotoxicity by stimulating the host immune system. In the Children's Oncology Group study AOST1421, the GD2 antibody dinutuximab was investigated in combination with GM-CSF in patients with recurrent pulmonary osteosarcoma who were in complete surgical remission. This was a single-arm phase 2 study. The study evaluated the disease control rate, which was defined as the proportion of patients who were event-free at 12 months from study enrollment, in patients with recurrent pulmonary osteosarcoma that were completely resected, treated with dinutuximab plus GM-CSF cytokine therapy versus the historical benchmark. Events were defined as progressive disease or death within 12 months due to treatment or progression of disease. The historical benchmark was Children's Oncology Group Protocol AOST 0221, investigating inhaled GM-CSF with first pulmonary recurrence of osteosarcoma. The 12-month disease control rate in that trial was 20%. In AOST 1421, dinutuximab did not demonstrate sufficient efficacy with a disease control rate of 30.7%. Success for this trial was defined as a greater than 40% event-free rate at 12 months from study enrollment.
Hey, Dr. Yoon, I had a question about one of the notes you had about GD2 expression. So you noted that you would see higher expression of GD2 in the kind of recurrence or the metastatic disease.
Correct.
And I'm curious, do you see it? So do you just see if someone presents with GD2 at, diagnosis that they have higher expression with metastatic disease? Do you see patients that don't present with GD2 at diagnosis, but then have GD2 in their recurrence?
That's a really good question. GD2 is not one of the markers we typically look at with in upfront osteosarcoma. Excuse me. And I think with the use of more, where we're looking at next-gen sequencing and we're looking at more molecular tumor profiling, and more of us are starting to send it at initial diagnosis and not just at times of relapse, I think we're going to get more information on that. I don't have an answer to that. And I don't think that we have investigated that. And I don't know how reliable that data is, just because previously we haven't been using next-gen sequencing as frequently as we are now. But that's a really good question. And I think that as GD2 is being examined as a potential target for osteosarcoma, that's definitely a place for more research to be done to really look at the different time points in treatment to see what the GD2 expression looks like? It's a really good question. And looking at the role of radiation therapy in osteosarcoma. So osteosarcoma has traditionally been viewed as a radiation-resistant tumor. And so therefore, it's been limited to be used for local treatment for unresectable tumors and for palliation of symptomatic metastatic disease. So as a way of background, looking at the GD2-SADA 177Lu-DOTA drug platform, radioimmunotherapy is defined as the combination of radiation therapy and immunotherapy. So in immunotherapy, a monoclonal antibody is targeted against a specific cancer antigen. In radiation therapy, as we all know, a radioisotope is delivered that delivers radiation to the targeted tissue. In the traditional one-step radioimmunotherapy, a monoclonal antibody is paired with a radioisotope. The antibody then binds to the target, and the radioisotope will irradiate the target. However, the dose is limited by toxicity as depicted by the red curve on the right, because there are these circulating long-lived antibodies which expose the non-targeted tissue, specifically blood and bone marrow, to high levels of radioactivity. So this study is investigating the SADA platform, which is a two-step pre-targeted radioimmunotherapy. SADA represents a two-step self-assembly and disassembly bispecific DOTA-engaging antibody system concept, which is referred to as liquid radiation. So there are two steps, as we discussed in the SADA platform. The first step is the tumor pre-targeting. So the SADA molecule, which is an antibody construct, is first administered and binds to the target on the tumor cells. So in this study, it's GD2. Within approximately 48 hours or longer, we're not sure yet, the non-bound SADA should disassemble and clear from the circulation. The second step consists of the administration of the radioactive payload, which in this instance is 177Lu-DOTA. When the free SADA is cleared from the circulation, the radioactive payload, 177Lu-DOTA, is administered. This payload will then bind to the anti-DOTA segment of the SADA molecule and irradiate the target. The unbound payload, the unbound radioactive particle, should then be cleared from the circulation within hours. So this is the SADA molecule construct. It's composed of three parts. The anti-tumor part, depicted in blue, binds to the target, which is GD2. The anti-DOTA part, depicted in green, binds to the radioisotope, the 177Lu-DOTA. The p53 domain, depicted in purple, allows the construct to self-assemble and disassemble. So here's a side-by-side comparison of one-step radioimmunotherapy versus the two-step SADA platform. As previously discussed, conventional radiotherapy is limited by the long-lasting antibodies and therefore causing increased toxicity to non-targeted tissues, specifically the blood and bone marrow. With the pre-targeted SADA technology, the two-step technology, due to the rapid disassembly of the SADA molecule, there is targeted radiation dose to the tissues of interest. So once again, the SADA platform two-step radioimmunotherapy binds specifically to GD2 and to the 177Lu-DOTA. There's focused irradiation of the tumor tissue with limited exposure to non-target tissue and blood, compared to the traditional one-step radioimmunotherapy. 177Lu-DOTA is radiotherapy combining radionuclide with a peptide that specifically binds to peptide receptors and therefore delivers targeted radiation.
Dr. Yoon, so just to clarify, so in that first step with the GD2 binding, so it's basically honing in on GD2, right, on the cancer cells. And then there's also then a GD2 antibody attached to that? Is it like a dinutuximab or something, or?
So it's an antibody construct that binds to GD2. So it's a different it's a different formulation than dinutuximab. So all of the anti-GD2 antibodies are formulated differently. And this one in particular has the SADA construct bound to the GD2 that's to allow the rapid assembly and disassembly, and then also has a component that binds to the lutetium DOTA. So it's a radioimmunotherapy construct as opposed to the traditional anti-GD2 therapies that we know of, the dinutuximab and the nexictimab.
Okay. But it still has that anti-GD2 component. So it's kind of, first step, it's basically honing in on the cancer cell.
Correct.
Bring that antibody. But then that also kind of plants the flag so that when the anti-DOTA part comes in, it knows where to go. Is that right?
So so the antibody construct has has GD2 and anti-DOTA. So it has both. And so what happens is that once the GD2-SADA is given, the antibody construct will bind to the GD2, and then the construct that's bound to the GD2 will remain in place. The unbound SADA GD2 construct will be cleared through the circulation, and then that allows the the lutetium DOTA, which is then administered roughly 48 hours later, to then bind to the antibody that's bound to GD2.
Okay, got it.
Does that make sense? I know it's very confusing.
I know. Does that make sense for you? So basically, GD2 goes in first, but then when the DOTA comes in, it binds to the GD2. It can.
So there's the lutetium DOTA will bind to the anti-DOTA part of the construct. So there's if you think of it as having kind of two prongs to this antibody, one to bind to the GD2 and one to bind to the lutetium.
Okay.
So it's a two-step in the sense that one will be bound to the GD2 on the tumor cells, and then once the lutetium particle is given, it will then bind to the DOTA component of the antibody.
Mm-hmm. But the GD2 goes in first, and that's kind of what's helping kind of identify the cancer cells?
Correct. It hones to the tumor cells of interest. Thereby, once the lutetium is given, it's going to be targeted irradiation to the cells that that construct is bound to.
Mm-hmm. So it kind of goes and finds the cancer cells, has the anti-DOTA thing on there so that then when the anti-DOTA goes in, it knows where to go.
Exactly.
Yeah. Okay. Thank you.
Yes. Thank you for the question. I know it's very confusing. It took me a long time to think this through.
The other question I had, just to clarify, because, we talk about P53 a lot, right?
Yeah.
And not just because it's a highly commonly mutated gene, but I'm guessing that doesn't have anything to do with this P53?
Yeah, it has nothing to do with this. It's just, it was part of the way in which this construct was developed, and it allows the rapid assembly and disassembly of this particle.
Thank you.
Yes. So moving on to the study design. So this is a first-in-human phase 1 single-arm, multi-center, non-randomized, open-label study, and it will assess the safety and tolerability of the GD2-SADA combined with the 177 lutetium DOTA drug complex. It's composed of 3 parts, parts A, B, and C, and the dose escalation is based on a 3x3 trial design. The study population is adult and adolescent patients with recurrent or refractory metastatic solid tumors known to express GD2, which includes small cell lung cancer, sarcoma, and malignant melanoma. The plan is to enroll 60 patients within the United States only, and the study is currently recruiting patients. So here's a schematic of the study design. In Part A, the GD2-SADA protein dose escalation will be investigated, and there will also be testing of dosing intervals between GD2-SADA and 177 lutetium DOTA. And then there will be a fixed lutetium DOTA therapy dose that is given. This is composed of 1 treatment cycle only. For Part B, the 177 lutetium DOTA therapy will be dose escalated and will be combined with the fixed GD2-SADA protein dose that was found in Part A. And patients can be on Part B for up to 2 cycles of therapy. And then in Part C, you can have up to 5 cycles of repeated dosing of the GD2-SADA dose that's determined in Part A and the lutetium-DOTA dose that's determined in Part B. And patients can only participate in one part of this trial.
Oh, I see. So you can't kind of graduate to the next step.
No. And I think that's really important to bring up because you can only be in one part of the trial. So if you, enrolled in Part A, you can't enroll in Part C later on.
And then Part A, one of the things you said you were looking at was the dosing interval.
Mm-hmm.
In one of your earlier slides, you had noted kind of the time it takes for the SADA molecule to unbind.
Yeah.
It's guesstimated at around maybe 48 hours. That's what you're trying to ascertain, is that?
Correct. What is the optimal dosing interval between the time the GD2-SADA is administered and when the lutetium-DOTA is administered? And so since this is a first-in-human trial, we don't know how rapidly the GD2-SADA molecule is cleared from the circulation. So that's why there's a testing of the dosing interval as well that's built into the trial. So the treatment cycles are composed of an imaging part and a therapy part. The first part, the imaging part, step 1 is the GD2-SADA dose. And the step 2 is the 177 lutetium-DOTA imaging dose. And so because this is the imaging dose, a SPECT CT scan will then be done to determine tumor targeting. We would expect the tumor to light up when the lutetium-DOTA imaging dose is given, since the GD2 antibody should be bound to the GD2 on the tumor cells. Only patients with confirmed tumor targeting will move forward to the therapy part. And for the therapy part, in step 1, the GD2-SADA dose will once again be administered. And in step 2, the 177 lutetium-DOTA therapy dose will be administered to result in targeted tumor irradiation. So due to work from our neuroblastoma colleagues, the anti-GD2 therapy adverse reactions are well known. The most common reactions are infusion-related reaction symptoms. And so these are treated with antihistamine, acetaminophen, corticosteroid, fluids, and antiemetics. Anaphylaxis can also occur, and this is treated with epinephrine and corticosteroid. And neuropathic pain is also a common adverse reaction, and this is treated with gabapentin and morphine. So the selected inclusion criteria, so for all patients for the small cell lung cancer, sarcoma, and malignant melanoma, it's for patients over the age of patients 18 or over. But for the sarcoma population only, patients who are 16 years of age and over can enroll in this trial. For sarcoma patients, they have to be recurrent or refractory disease after receiving at least one prior line of therapy of standard systemic anthracycline-containing therapy. You have to have measurable disease according to RECIST 1.1, an ECOG performance status score of 0 to 1, expected survival of over 3 months, platelet count greater than or equal to 100,000, hemoglobin greater than or equal to 9, and absolute neutrophil count greater than or equal to 1,500. You also must have adequate renal and liver function. So selected exclusion criteria, systemic chemotherapy, radiotherapy, immunotherapy, or any major surgery administered within 3 weeks prior to the first planned dose of the investigational medicinal product. Patients receiving any other investigational therapy for their cancer within 3 weeks prior to the first planned dose are not eligible. Any ongoing radiation toxicities from prior radiation therapy. Patients with a diagnosis of autoimmune disease or immunodeficiencies or documented infection with HIV or active hepatitis B or C. Any prior treatment with anti-GD2 antibody therapy is not allowed, and patients must have a body weight of greater than 45 kilograms.
Dr. Yoon, is radiation therapy included in that washout? Is that different from the radiotherapy?
Yeah, so any kind of radiation therapy, it has to be a washout period of 3 weeks.
Okay, and what about? Oh no, go ahead.
And you cannot have any lingering radiation toxicities.
Okay. What about interventional, like cryoablation or ablation, different types of ablation?
So I think it's I would still believe that would be part of the 3-week washout, just because of, any cancer-directed therapy, whatever it may be, it should be within 3 weeks. But with all the screening that needs to take place to even enroll on this trial, once a potential subject is identified, by the time you go through all of the screening and the eligibility checklist, it's probably going to be close to about 3 weeks. So, but any cancer-directed therapy should have a washout of 3 weeks.
Okay.
So the primary objectives in Part A is to determine the optimal safe GD2-SADA protein dose and the dosing interval between GD2-SADA and the lutetium-DOTA administrations, as we previously discussed. Part B will be to determine the maximum tolerable activity of 177 lutetium-DOTA, the therapeutic dose, which is different than the imaging dose. And Part C is to assess the cumulative toxicity signals and safety profile, so the number and severity of the adverse events following repeated dosing, and determine the recommended phase 2 dose. The primary endpoints or the occurrence in Part A is the occurrence of dose-limiting toxicities with the GD2-SADA construct. In Part B, it's occurrence of the dose-limiting toxicities with the 177 lutetium-DOTA treatment dose. And Part C is the number and severity of the adverse events. So the sites that are currently enrolling patients, Honor Health in Scottsdale, Arizona, my institution, City of Hope National Medical Center in Duarte, California, Memorial Sloan Kettering Cancer Center, and UPMC Hillman Cancer Center in Pittsburgh, Pennsylvania are currently enrolling patients. And the first patient has been dosed in April. And I'm happy to take any other questions, and I couldn't help myself. I had to put this in there. May the 4th be with all of you today.
Thank you for that. I forgot about that.
It's been mentioned to me so many times today. I had to say something.
Well, one of us had to, so I'm glad you did.
So thank you.
Okay, well, I think you did a pretty thorough job explaining the trial, Dr. Yoon, so thank you so much. Appreciate you taking time to join us on OsteoBites today and making it better for osteosarcoma patients and really all sarcoma patients. More information on this and all OsteoBites can be found on YouTube, on our website at mibagents.org, and at your favorite podcast place. Please join us next week on May 11th. We're going to be talking to Dr. Lee Hellman and Chelsea Blanks. Dr. Hellman is the director of the Osteosarcoma Institute, OSI, and Chelsea Blanks is the OSI Connect administrator. And they're going to be discussing OSI Connect, which is the Osteosarcoma Institute's free, easy-to-use resource for patients who have been diagnosed with osteosarcoma or suffered a relapse after initial treatment. And you'll learn about how OSI Connect works and examples of how the program has helped more than 70 patients since its launch in 2019. And you can find the rest of our OsteoBites lineup for the next few months on our website. If you have any ideas for future topics you'd like to hear about, please share them with us at events@mibagents.org. Thank you again, Dr. Yoon, Vicky, and Walker, and for all of you for spending an hour with us today. And we hope to see you back here on OsteoBites next week on May 11th when we talk to Dr. Hellman and Chelsea from OSI. Thank you, everyone.